Okay, so hello everybody. I think we can slowly start. We will have a few minutes introduction, so the people that are still joining will have a few more minutes to join. So hello everybody. I'm very much happy to welcome you here to this next seminar series from GloBIAS. Today we have a great speaker, Nicholas, and he will tell us about big data infrastructure strategies for image analysis in a core facility. This seminar is part of the next topic that is focusing on data management and optimization of data flow between different devices, sites, and stakeholders. My name is Eva. I'm joining from Prague, and I will be chairing today's seminar. So in the next few minutes, I would like to briefly introduce Nick. Then I will tell you a few things about GloBIAS, and then I will pass the words to our speaker. So let me tell you about Nick. He is joining us from Australia, where he is working at the Advanced Imaging Platform. He's employed as the Senior Principal Imaging Scientist at the Institute for Molecular Bioscience at the University of Queensland. He finished his PhD in 2018, where he was focusing and studying the macrophages, their behavior, and he was using the novel life cell imaging techniques. And since then, he's been working with many expert techniques in light microscopy, including, for example, light sheet or high throughput screening. And he's been also developing various image analysis workflows. Moreover, he's an expert that is collaborating with many other experts from many various areas, and he's focusing on development of big data management tools and infrastructure for the high performance computing. And one specific tool that I would like to mention specifically is the image processing portal, right? Which is something that Nick is involved in. And his work on big data was supported by John Zuckerberg initiative. I'm very much looking forward to him sharing his work with us. But before I pass the word to Nick, I would like to say a few words about GloBIAS and the opportunities that we are offering. So as many of you probably know, GloBIAS is the Global Bioimage Analyst Society, and we are trying to connect bioimage analysts from all parts of the globe. If you are interested, you can follow us on LinkedIn and also on Blue Sky. And if you are interested, you can also become a member and get involved in the different activities that GloBIAS is involved in. One of the activities that I would like to mention is the next free help session. This is an event that will take place between the 20th and 24th of April. And the idea is to share the expertise in bioimage analysis that GloBIAS has with many researchers around the globe. The idea is that the researchers or image enthusiasts can apply with their image analysis questions, and they can get individual sessions with people from GloBIAS that will help them with the question and guide them, for example, to select a specific tool or approach to tackle the image analysis problem. So if you guys are interested or you know somebody that might be interested, please spread the word, because I think this is a great opportunity for people to get help with image analysis. A next activity that GloBIAS is involved in is the seminar series that you are attending right now. Seminar series is a dynamic platform for bioimage analysts that is attempting to give them an opportunity to stay up to date with the latest development. And with that, I would like to thank the Broad Institute and also the University of Helsinki for providing the infrastructure. And I would like to also shout out to Elnas and Beth that are actually the masterminds behind this activity. And just to notify you that the seminar is recorded, and in a few days it's going to be posted on YouTube. And also we will attempt to answer the questions that will be asked later in the Q&A, and we will try to post it on ImageSC again in a few days. So I think with that, I can pass the words to Nick, and I'm very much looking for the seminar. So I stopped sharing. So Nick, I think you should be able to share your screen, and the floor is yours.
Thank you. Hopefully you can see my presentation.
Yes.
Thank you, Elnaz, Eva, and the GloBIAS Society for the opportunity to speak tonight. So my name's Nicholas Condon, and I'm coming from Australia this evening, highlighting, I think, the global nature of GloBIAS. So tonight, I'll be talking to you about the big data infrastructure strategies for image analysis in a core facility, or how we're doing it in our core facility in Brisbane. I know. Can I do it this way? My PowerPoint does not seem to. There we go. So first of all, I'd like to acknowledge the traditional owners of the lands in which I'm coming from tonight, which is the Yagara people. I pay my respects to their ancestors and their descendants who continue cultural and spiritual connections to country, and we recognize the valuable contributions to both Australian and global society. And the Aboriginal people of Australia come together and learn about and share knowledge for thousands of years, and we're continuing this tradition tonight. I just wanted to briefly start with where I'm from. I'm from the University of Queensland, which is on Australia's east coast, about halfway up in Brisbane. We're a university that does teaching and research and is often ranked in the top 50 around the world. Where I work is this building, the Institute for Molecular Bioscience, and we have 36 research groups housed in it with over 500 researchers. We have about 170 PhD and a higher degree research scholars, and with about 100 or more industry partners and spin-outs. Collectively, the Institute operates under kind of 4 centers, whether it's population and disease genomics, superbugs, cell biology and chronic disease, or chemistry and drug discovery with all the lovely insects that we have from our country. Collectively, these all kind of funnel into a number of outputs. But the take-home message here is in our facility, I get to work with really disparate things, whether it's working with industry on agricultural products, whether it's in disease and health, whether it's in microbes, antimicrobial resistance, endometriosis, pain research, all different lovely things, which is great for a microscopist. Our facility consists of currently 3 staff. We're hiring but probably only locally because it's a short-term position. But we have 20 microscopes and 20 servers that I'll talk about later, and we serve about 200 users annually. We conduct, about 160 microscope trainings, and that's over about 55 different projects. I was introduced before where it was announced that I'm a cell biologist by training. I am not a computer scientist. I'm not a developer. A lot of these people photographed here are the different teams, people from all different areas across the university, both past and present, who have contributed to the different stuff I'm going to talk about today. And so I may end up deflecting some of your questions to those people, the specific teams. And we do that all the time because people tend to like to know what we're doing and how we do it. But in our core facility, we've got lots of microscopes. They're all light microscopes. But I thought I'd kind of show some of the different things that we do. And so we have quail model organism researchers. They use confocals and stereo microscopes. We have kind of slide scanning, tissue, spatial people, confocal with developmental biology, spinning disk for developmental and high-speed imaging, and the lattice light sheet for cell biology. So already, hopefully, you can appreciate there's a vast difference in what we're kind of doing. And some of the things that the data sets that these systems produce are really different. So we might have kind of big tile scans, multicolor time lapse on some systems, where others we have just 3D tile scans for cell slide scanning, developmental biology on our confocals, but thankfully because they're point scanning, they tend to be pretty size limited. The spinning disks are the kind of somewhat scary systems because they're turnkey and can make terabytes of data. And then we have like our lattice light sheet, which we have the 3i system, which is, it's a bit scary looking for a lot of the biologists. So it becomes kind of a bit of a hand-holding system. So it's not too bad, but it does produce a lot of data. And it was the impetus for a lot of the stuff that I'm going to talk about today, because it was the system that we got that kind of began this big data journey. I like to think that we've solved storage. It's relatively cheap, fluctuates a bit with the market, but the university has a really good system that we've helped shape and develop with various different teams over the years. The problem now that I see is the flow, which is kind of the theme of this seminar series, getting the data to these different the analysis steps and sharing it. So at the University of Queensland, we have a petabyte scale storage system that's called Research Data Manager. It's a one-stop shop for the researchers to manage all their data that meets the FAIR principles, as well as all the relative governance and risk and compliance standards for Australian universities and research that's conducted in Australia. It supports multiple logon methods where we can have industry partners or other people from around Australia or the world log on with their credentials and have access to the system. And it's project-based where we can link in a whole bunch of other things, not just the storage, where we might have animal ethics associated with it, human ethics, the grants and funders, digital research notebooks, our government field of research codes, and soon we're linking in the LIMS or laboratory inventory management system with this thing so we can start to really tease apart for one project how much data is produced, what kind of data is being produced, what reagents are being used. We can link it with the booking of the instruments and see, does X government funded project use how many hours on a confocal and therefore how much support and things. So we can start to really mine this for a lot of data. The way it works is the university's partnered with two major computing suppliers, IBM and HP Enterprise. And we have what we call the kind of custom data fabric. That's just because it's a bit of a conglomerate of both tools. But the way it works is we have lots of IBM storage, whether it's depending on the generation it was purchased, Spectrum Scale, GPFS, the ESS for elastic storage. And then all that's managed by a product called the DMF7 or Data Management Framework 7, which is the HP product. That's really complicated jargon, but what it means is that each collection gets its own identifier code, and that's linked out to that project stuff I was talking about, and we can link that then to the booking systems and the instruments and things like that. It's a tiered storage infrastructure that's housed at multiple locations. So we have high-speed storage that we call like the hot active, and that's near the instrument, so that's on campus. And then we have warm and colder and tape backups that are kind of off-site. And the system means that users don't have to worry about where the data is. That's all managed by this data fabric. So if I'm accessing data on a microscope in one building, and then I want to process it on a visualization server that's in a different location, the data appears there and all the movements and migrations are managed automatically. So users don't ever have to worry about how much data they're producing, how much they're throwing in. They don't have to worry about whether it's backed up, whether it meets the requirements for their grants and things like that. That's all managed centrally. The collections that we have can be connected and sent out to the world. So we can flip these and make them public and mint a DOI through the library's ecosystem. And we can share those data sets part of publications. But obviously, it's better if we can put these into the more public repositories globally. We can send data out through transfer methods like GloBIAS. We can link it in, send it to an AWS bucket, or we have in Australia our own research fiber black network between all the universities that we can make access to. So hopefully you can appreciate that this whole storage of the data is what I like to think pretty well solves on our end for our users. My users probably don't think the same way though. And that's kind of great because if you're a computer scientist and you deal with all the command line and stuff, it's really nice. We have got some tools for Windows to make it a bit easier so they mount automatically on microscope computers for them and things like that. But we do need to kind of keep building tools that will address this movement of data, making sure the infrastructure is usable and that it's reproducible. So we build a lot of tools, whether they're just simple little PowerShell scripts that run on microscopes to whole kind of platforms and web infrastructures that we build. But we kind of try and follow these major design principles, which is we don't want to move the data, right? I've already said that's the painful part. So once the data gets into the system, we don't want to be copying it back and forth out of the main storage to computers or things like that for analysis. We want to leave it there and access it to it. We want to minimize duplication, right? We have users making terabytes of data every time. I don't want them to have 5 copies of that and then each of those 5 copies getting backed up everywhere. We get that researchers are really busy. They don't want to learn all different environments and things. So we try and keep everything really standardized for them so that it's familiar and it's kind of easy to use. As a both kind of, I'm going to say, ex-researcher and current core facility staff member, I know that if you don't capture the metadata, and I don't mean like the objective and stuff, I mean like how was the sample mounted, what drug was added, what was the batch number. If you don't do it early, it's not going to happen. So how do we make tools that can kind of promote that early on? And where possible, because I only have 3 staff with us in the facility, what can we automate to save time? And I'll show you today the different tools that we're building, and kind of hopefully you remember that they map back to these principles. It should come as no surprise to this audience that, the typical flow that we see in our core facility is someone has an experiment, they then want to image it, then they need to do some analysis and visualization, and hopefully it gets shared and published. So we as a core facility are trying to build stuff to speed up this flow. We don't like to stop the science, is what we say. So there's bottlenecks that we've seen all the way through that pipeline. And I don't like to think that we've addressed all of them. These are just some of the things that we're doing that we've seen really positive benefits from. So I see at the microscope, the computers that the vendors give us are usually pretty junkily spec'd. We see pretty poor user behaviors that we can try and address, and that capturing metadata at the microscope is also something that we think is quite important. So at the microscope, the computer you buy, it's going to look something like this. It might be some Dell workstation, it might be some HP workstation. It's going to be pretty chunky to begin with. But usually the first thing we do is put it on the network and make it so that every user logs in with their own credentials. So there's no admin accounts for the users. They just log in with their standard university log on, and that helps us track who's using what, so we can get a bunch of stats. Are the cell biologists using this system? Are the developmental people using it? Are people kind of shifting with trends? It also means that we have consistent settings and layouts. So when one user logs in and they change something for their needs, it doesn't affect the next user, and then that user isn't coming and finding me and going, something's changed. So that kind of frees up my time. And it means that because they've logged in with their university credentials, we know who they are. We know that they've been authenticated, and we can map all the different RDM projects that they might be on automatically to their session, so they don't have to go typing in long network addresses and things. Another thing we do is we add NVMe rated storage cards. We buy these ones called Highpoint. They come from Amazon, and they're probably under, say, about 500 USD. This allows us to put in something like 32 terabytes of storage. Obviously, that doesn't include the storage cost, but it lets us get 100 gigabit I/O. So we have hard drives that we can be siphoning off data to the network and the microscope running and pumping data into, and we don't see any issues or crashes and things like that. So that just means that users don't have to wait to the end of their session, and the data that's moving off is not speed limited by the hardware, because a typical microscope might come with some SSD, or maybe it's got a small NVMe stick, but for our use case, usually that's not enough. And we have a whole bunch of scripts that we deploy. I've got a link on the last slide with the GitHub repository, but we have auto delete scripts, so I don't have time to go to every microscope and delete stuff. It's programmed so that it won't delete configuration files and things like that. It does go hunting if someone wants to store something deep down in some hidden folder on their account, it'll still delete it. If it can't delete anything, it sends us a little email or a Teams message going, help, I can't delete, you have to come and help. So that frees up time, and we maybe get like 1 or 2 of them every couple of months. So we don't have to go to the microscope every week and delete files. We also use the Stratacore PPMS, and we have a little floating, you can report incidents, or you can book extra time and things like that straight from the microscope session. You can take screenshots and all that. But we can't optimize for things that we can't see. I can't be in every scope room looking over people's shoulders. So we want to be able to like look at what's the strain on the computing hardware. Where do we need to spend money? Because that's always, a contentious issue. We don't have a lot of it, so we need to be spending it wisely. Are we seeing issues with transfer speeds? And maybe we can start to think about what's being imaged on each system beyond just going, oh, that was so-and-so and they're in this group and they're probably imaging a fish or something. So we've written a script that pushes to a Google Sheet in the cloud, and soon we're going to change it to a C table, but all information about every session. Every minute it logs it does a check and says like how much RAM is being used, when was the last time the mouse was used? So that tells us like, are people actively at the instruments or are they kind of setting up things and going away? And we can then mine that to say, well, is something always maxing out or is it not maxing out? And this becomes really important when we have our virtual analysis computers because we can kind of reshape them relatively easily and make more of them or combine ones if we're seeing changing user behaviors. So yeah, is it at the microscope? And like I can catch like, are people running analysis on the instrument computer, or are they kind of, the software is not running, but they're logged in, so they're probably transferring data. Do we need to modulate some things there? But I've also been kind of following the REMBI, and I'm part of Founding Guide. And I'm conscious of the fact that we need to be capturing kind of more metadata that's not just the acquisition, because that's not the whole context of the experiment that's being done. So a lot of our instruments, like I'm guessing most people's, might still be sitting on Windows 10. I think I have one on Windows 7, where we can't access the web, where we have our digital notebook interface. So that can be kind of difficult as well, just accessing the outside web from inside the building, or inside on the instrument network. I also, as an image analyst, see some really poor naming conventions, image 1, 2, 3, 4, no descriptions, nothing really provided. And so, as I mentioned, Founding Guide, Core Epilimi, REMBI, all kind of providing guidance on stuff that we need to start capturing. And I've had, the group leader come to me and say, I've uploaded this paper and they want this data put somewhere and I don't know what this means. So we need to start capturing this data early. And I've begun kind of playing with making a tool for that. So can we capture information about the experiment and make it interoperable to the digital research notebook? So we use the lab archive infrastructure at our university. And can we also kind of prompt the user to describe, can we squeeze out some more information by kind of help coaxing it out of them at the instrument? So what kind of sample did they have? How is it labeled? What project does it belong to? Because if they say it's from XYZ projects, now I know what grant funded it, whether there's ethics attached to it, and a whole bunch of other information. Who was the PI? All this stuff that kind of falls into some of these categories that REMBI is guiding us to say that we need to be collecting. So I built this thing purely vibe coded, but it's a little GUI that runs, it's a Python GUI that helps users prompt get their data in. It can spit out things to text files, XMLs or JSONs, or tab delineated for Amero. And it kind of gives a human readable and machine readable metadata tool. This is what it looks like. It's not super pretty, but it helps get some information. And we have this running on all the instruments. Actually, what it also does is researchers are lazy, so we could capture some of the instrument metadata as well by just loading a file, and I leveraged some of the libraries for CZI readers, LIF readers, things like that. And they're not perfect because there's so many variations across the different systems, and we're still running some 710 Zeiss systems, for example, that are getting a bit old. So, but it mostly works. A couple of things that happened that I learned, we could make it work both ways so that you could load up a template file, because as I said, researchers are lazy. They tend to do the same thing over and over. So that was nice. Getting people to use it, I found that because it was searchable by the operating system, a lot of the group leaders, a student might come in for a little while and then leave, and they might know that they were working on a certain confocal. They might know that they were using a certain dye. All that information searchable in like Mac Searchlight, Spotlight, or Windows Explorer. And that became the kind of carrot to say, well, if you can convince your users to use this tool, then they might they'll use it because you're telling them to do it kind of thing. But it's still kind of pretty lightweight and janky. Can we use the info here to schedule transfers? So the computing center has worked with the EM facility where when you book the instrument, you can use this tool called Pitchy, which is a Clouder instance that they operate, and says, you booked it under this project, so all the data gets hoovered up into that project automatically. So we could build that into our pipeline by knowing which project they used. I want to move away from free text fields because that's a horrible idea I've learned. The amount of variations of just GFP I see. So we need to move to ontologies. But also as a facility, I can now start to mine it and go, well, is anyone using this objective on that system? Maybe if they're not, we could take it off. It's less likely to be damaged. We can look at usage patterns in a bit more detail. But microscopy data should be looked at, and for this audience, quantified. But that can be really hard in the age of big data. So we also have spent a whole bunch of time tinkering and playing with virtual servers. To the extent now that our university does not allow you to buy a workstation PC, it must be a virtualized server. These are providing a really viable solution to the big data problem. The reason being is that they're servers, so they have 100 gigabit network cards, are going to live in a data center that have those high-speed networks, hopefully close to some of those hot active caches that we have. They usually can support way more sockets and therefore CPU cores. They support enterprise GPUs, which lets us do some really funky stuff, which I'll talk about later. And they have lots of RAM. Can be difficult in this climate, I know, but it means that we can have, multiple terabytes in a single VM. And they can be accessed from anywhere with relatively low bandwidth. This is quite an old photo, but colleague and GloBIAS member Lachlan at WeHi, we had a bit of a competition back in the early days of the stupidest places we could do image analysis remotely. But we've been doing this for almost a decade now. So moving the servers into the data center has been a game changer for us because it just gives a way better user experience for the workflows interactively. We can do consistent environments because it's all virtualized. We have the same desktops and all the software gets managed nicely to an extent. It's remotely accessible. This was great during COVID, but also just in general. And I'll touch on some things that we've noticed there. It's really scalable. And I'm going to say it's easily supportable. We kind of have stolen about 0.1 of our IT, one of our IT team's experts who helps manage some of it. But once it's set up, it's pretty easy to leave and have running. We run mostly static VMs, so they're not dynamic on the fly. But so I wanted to touch on how these can be configured because I thought this might be useful for this audience. So one server or physical host can become multiple VMs, so you can carve it up. So in this scenario, I've just picked some random specs, and you can take that and kind of split it into 3 equal machines, and that's pretty straightforward. But you can also carve them up differently and asymmetrically. So you can have, here I've adjusted the size just so the math was a bit easier, but you can have one bigger or medium virtual machine that might have 32 CPU cores, a terabyte of RAM, and 1 whole enterprise GPU. And then we can have 2 smaller ones so that we can kind of load balance the workflows that people use, right? So we might have someone doing a lot of light sheet processing, so all the light sheet software is on the bigger box. And then if it's kind of confocal work, we might have our offline Zen and those programs on a smaller VM. But we can also virtualize the GPUs. And this is something that when I discuss with people that they're not really aware that this is possible. So with the enterprise grade cards, these aren't like your NVIDIA 5090 Ti cards. These are the enterprise GPUs. You can use a platform called, or kind of like a driver called Virtual Workstation. It used to be called Grid. And this lets you chop up the GPUs. And so you can take 1 GPU and then present that as multiple smaller GPUs. And this is shamelessly stolen from NVIDIA's documentation. The amount of money we've spent with them, I'm sure it's fine. The in the example here, we have a VM that's receiving two kind of chunks of a single GPU virtually into it. And then we have a second VM that's receiving four virtual GPUs, but they're actually coming from two different physical GPUs. And the reason that you pay, because this isn't free, is you get this management layer that lives in the virtualization layer. But as far as like the chopping up, it's relatively straightforward. There are some limitations. So, as always, it's not perfect, but it does cost money. It's not that expensive. I've said it's about $1,000 Australian, so maybe like 600 euro. And that's usually for a few years. We in Australia have kind of socialized. All of the universities have a kind of, we pool our money and buy the big buckets of licenses and then divvy them up. So if you are going down this path, maybe check that you're not buying something that already exists. And another kind of limitation is that there's defined sizes. You can't just pick whatever you want. So across the kind of more recent GPUs listed down here are the different shapes you can do. And a physical GPU can only be carved up the same way. So what I mean by that is if you've got a single L40S that's got 48 gig of VRAM, you could have one 48 gig virtual GPU presented, two 24 gig, but you can't have like one 24 and then two 12s on top from the one card. But what you can do if you have multiple of these is have one carved up into some big ones and then one carved up into a lot little ones and then present all of them into different VMs. And not all of the GPUs that are enterprise will work for this. So something like the A100, that doesn't have virtualization. It's only got compute. So just be aware of that. But you can use this to make even further non-symmetric VMs. And this is kind of based on one of our hosts that we do run concurrently at the facility or the platform. So we have a single host that's got 96 CPU cores, 2 terabytes of RAM, and 3 of these L40S GPUs. So we have two, what I would class as like a medium VM with 24 CPUs each and 256 gig of RAM, and they each get one whole GPU. And then we have 8 smaller ones that run a lot of our open source software, but we do give them some GPUs. They get one card carved up into 8 by 6 virtual GPUs. And that's kind of enough just to kind of run a small cell pose job or, beginning dabble with some of these machine learning tools that are becoming more prevalent. Whereas the bigger GPUs allow for some high memory deconvolution, so you can load in like a whole large volume to process without chopping it up. But we see lots of benefits to moving to, I say, the cloud. It's what we call our edge cloud. But the main thing is this resource flexibility. So we can clone VMs, which is great because I don't have to those 8 VMs that are small, I can just build one, put all the right software on, and then make 8 of them. And this is really good when we want to run like a workshop. So we've hosted both commercial and open source workshops where we get all the licenses, get all the demo data, and just clone it and have lots. There's less complaints about access to resources, so we're not space limited. Previously, we kind of had 4 big workstations, and that was at 4 desks. And desks can be kind of a hot topic when we have 500 people in the building. So users can access from their own laptops where they are. We do have a couple of kind of thin client computers with a big screen because you do sometimes need a big screen, and I wouldn't advocate doing lots of image analysis on a 16-inch little laptop screen. But one kind of challenge is we've got 20 VMs. Once you've made them all, because they mostly are static for us, it can be a bit difficult with version control because you might have to go and update 20 different versions of Fiji. But we also cost recover on our VM, so that means we charge for access because we do the resource itself costs money to buy, and the licensed software that we have, costs money to maintain. But also, I did some quick analysis for a couple of years ago. This is in Australian dollars, and you can see that income by outside of hours. So we're seeing, 7,000 or about, 25%, say, of the income being generated outside of business hours. So I think that's kind of promoting, obviously, how much it's being used. Another cool thing we can do is we can oversubscribe our VMs and the resources. So what I mean by that is we can turn on and off the VMs as we need them. So on the kind of left half here is what I showed you. That's what lives there all the time and is running, right now. But I have 8, I'm going to put spare VMs sitting in the wings, and they just turned off. So if I want to run a, session on how to run CellPose in Fiji and have users actually be able to do it, and their laptops might not have discrete GPUs, I can book out and turn off these 2 medium ones, and then just turn those ones on, and I can have 16 now. Some of you may note that I'm leaving resources on the table here because I'm only giving 6 GB GPUs, and theoretically we could have 9 per one. There isn't a 9 gig profile though, but I could have 8. But the limitation there is when you host these computers, you want to use the NVMe drive that's in the server itself. You don't really want to be hosting the operating system from some network location. It gets a bit laggy. And we're limited to the size there. So I don't bother giving every single resource in the box for these workshops, just because it would be too much of copying the virtual computers down to the drive. There are other considerations. There's probably a lot more. I've only mentioned some that kind of were painful pinch points for us. Is licensing's a big one. The amount of software licenses that still require a USB and servers don't have a lot of USB ports has been a challenge. Straight up, some of the software won't run on Windows Server. And do you need licenses for the hypervisor and how you're going to connect in and those tools? Another important consideration is access control. If you're running a workstation PC and someone's booked it and someone else is sitting on it, it's a physical thing that you go to, you can tell them to get off. I don't know who's currently occupying a VM because it could be from anywhere in the world. So we have tools that we've written that links into the booking platform so that if you don't have a booking, it just boots you off. And then how do you limit the number of users to that thing? So we use Active Directory, and we have whitelists and that so that, someone from not our facility doesn't just get carte blanche access to the VM. And training. It did take a bit of a significant uplift for our users to understand what a virtual computer is, how to connect to it, the implications of logging out versus just closing the window. So that was kind of something I didn't expect, to be honest, that we had to teach, but we've made a lot of content around that. But we have also got high performance computing for when the data is too big. And we have kind of 2 access layers to this. We have on Bunya, our HPC cluster is called Bunya, and it's a play on Open On Demand. That's a really flexible platform and tool, whereas we also have our image processing portal, which provides a bit more guided or hand-holdy kind of experience for our users. I don't think this audience probably really needs this slide, but it's needed because we've got a bunch of camera-based microscopes and they make a lot of data. And this is like a mud map of some of the different resources around campus and how it all connects together. And I had that picture of the crying child in the gutter. We need ways to help users move the data seamlessly, and, getting it in and out of scratch on the HPC and stuff is not trivial. So also the other reason this is needed is this one's free because it's centrally paid for by the university. So our users like using it. And there's obviously bigger cameras and scary instruments like a light field coming. On Bunya is our interactive HPC processing task tool. It allows you, if you're not familiar with the Open On Demand infrastructure, it lets you create jobs, execute things. You can delete stuff all from the browser. It's got a relatively nice GUI. You can spin up virtual or dynamic Linux desktops, and we've based it on CVL, or the Characterization Virtual Laboratory. which is kind of a predecessor to, I think it's Bard, that EMBL is running. And you can also have Jupyter notebooks, you can run VS Code in the browser, and you can run terminals and stuff. And it has this really cool thing that lets you plug in your job ID number, and it tells you all about it and says, you asked for this much resource and you only use 10% of that, and can coach the users, which I think is really useful. But a lot of our biologists and that don't know how to spin up a dynamic Linux desktop with how many virtual CPU cores and virtual GPUs and that. So we tried to make a simpler tool, which is our image processing portal. It follows that similar philosophy of it's easy to use. It lives in the browser like the Open OnDemand platform. And it lets the users navigate around all the files, can move them, copy them, delete them, rename them, all that stuff on the server side. So they're not like pulling stuff through their laptop back out to some other location. We've built things for file conversion, deconvolution, stitching, and macro execution, and it uses like wizard-like workflows to kind of drip feed, get the information from the users, and then it can provide a receipt, which is, I think, really important. So if this plays, yep. This is just a quick video. I've shown this a few times around the world, so you may have seen it before, but this is just navigating our file system so I can see different things. I can go to launch a virtual desktop, and I've just given them the option as kind of small, medium, large, and then it's running. And I can kill that from the browser. I can go to deconvolution and choose what method and algorithms to use, go through a bunch of steps, get information. I can pull code from GitHub, or I can load up the files and then execute them. I can see what jobs are running. I can have some background configuration tools as an administrator and choose like the different deconvolution engine. We have got Huygens running in there too in a kind of test environment. The cool thing is though, because it's a HPC job, they've provided all the steps that they did, and it's in some, XML or JSON file that is used to create the job, but we can then put that into a kind of pretty table that the users can read, so they have a record, but we can then also use this to reconstruct jobs, and so like reapply them or like reuse the settings. And Slurm will store all of like the output files and things so that if there's any like log stuff that's needed, that's all kept as well. So the nice thing here is we know what was done to all the data, and it lives with the data in its form. And so if they've done at the instrument, used the tool I showed earlier, we know a lot about the sample, we know about the project information, and now we know what was done to it. And we've got a bunch of our users we've convinced to use and process it. This didn't happen overnight though. Younger, happier Nick went to Genelia back in his PhD, used the lattice. We came back and we bought one a number of years later. In that time, when I came back with a suitcase full of hard drives, we formed like the big data problem group. So we had people doing preclinical high resolution PET scanning. We had the serial block face EM and cryo EM coming. So we knew that there was this data avalanche. It was probably about late 2016, early 2017 that the first version of the Research Data Manager came out. After buying the lattice and installing it, we built our first version of the virtual desktop infrastructure. We had our first HPC cluster that's now being decommissioned. Our first version of the portal was back in 2019, so that's 7 years ago now. And we've updated it, bought more hardware, got a new HPC cluster that's now being kind of added to, and version 2 of the Research Data Manager debuted a couple of years ago with a few more features. Where are we at and what are we doing? So we have a path towards kind of automated data management from the instrument right through to the analysis and publication. I've got a kind of proof of concept tool for capturing some metadata. I do want to make some changes to it. We've got solutions for various levels of accessible computing or like interactive computing infrastructure. And we have the scalable centralized workflows with mechanisms to reproduce those for the processing and analysis. And all of that's resulted in like a pretty significant reduced support burden, so we can get by with 3 staff. But I still want to solve the issue around capturing the analysis steps being done in an interactive session. So like if someone's running Fiji on one of our Windows VMs, we are blind to what they were doing. And I need to link in the ontologies and stuff to the tool I showed so that we don't have free text fields. Lastly, very quickly, I've shown a lot. This has been a really long journey. I wanted to show 3 simple things that you could do in your institution to help kind of begin stress tests and figure out where your pain points might be. So stress testing your storage, mapping out your network, and measuring the resource demands. You can there's many different hard drive benchmarking tools. We use Autodisk. It's pretty good at showing us if we're seeing any issues with the hard drives. Even if you don't do that, if you're running Windows, you can bring up Resource Monitor, go to the disk tab, and look at the queue length. And if that number is growing, that means you've got tasks waiting to be done and your hard drive's not fast enough for what you're pushing it to do. So that's one way to check if that's a bottleneck. You can run tracert or traceroute. This is a command line tool where you can, from the microscope, say, connect to this storage location. And that will time how long it takes to get to each router or switch along the journey. What we found originally was some of the data was taking like the scenic route around campus a number of times before going out to the server. We worked with the IT and network teams to kind of better route it so that we wouldn't have that and saw immediate benefits. It's also, you get a lot more clout when you rock up to IT and you can name their switches and servers by name. And then lastly, we use this tool called GPU Profiler. It's developed by someone who works at NVIDIA and it's on GitHub, but it lets you run it while you do some task and it's graphing the CPU usage, the RAM usage, the GPU usage. And while, yes, you can do that in Task Manager, this lets you export it as a CSV, so you can kind of do comparisons and get the actual numbers. So that's been really useful for us. Lastly, there's a bunch of people who've been involved, both still with UQ and have left UQ, then a whole bunch of teams who have so many unnamed people in them, and all the researchers who've kept whinging and banging down my door saying, we need to go faster, I need to process this. I've received previously funding from CZI. I'm part of a number of different federally funded projects. We have OzBias, which is the Australian group, and Light Microscopy Australia, and plug that we've got this special edition on HPC and cloud resources that Beth and Rocco and I are editing. And I'm happy to take questions. And yeah.
Thanks a lot, Nick, for a great presentation. So I think we have like 10 minutes for the Q&A and we have more than 10 questions. So maybe I can start going through them and then the rest we can leave for ImageSC.
Yep.
So the first one is from Richard. He's asking about your experience with the division of the GPU. And he was asking whether most software would benefit from multiple GPU instances or whether you would recommend a single GPU per the virtual machine.
Yeah, it's a very good question. So not many programs going to make use of multiple GPUs. We are working with a couple of the common household names to be testing stuff for multi-GPU usage. Deconvolution was our main drive, where Microevolution was one of the first to have multi-GPU support. Also running that GPU profiler tool, you quickly learn that most stuff is not optimized, whereas you'll see a program just go straight to 100% and utilize everything of the GPU. But yeah, to be honest, most don't need multiple within one VM.
Then there are a couple of questions about the virtual workstations. So there is somebody is asking about the booking system, like whether you book a virtual machine or whether there is a queue system. There is also a question about which software you use, so whether it runs on Linux or Windows, and what exactly the setup is, and also what is your decision making about why exactly did you choose this setup?
Yeah, happy to answer that. So the virtual machines are, as I said, they're static, so we just kind of build them and then leave them. They are we run Stratacore PPMS and we just name the VMs by planets and Greek gods. and you book out that instrument, you get that instrument for the time that you have, and the licensed software that lives on that stays licensed there. So they are IP locked to the machines. Um, the So yeah, you book the instrument, you get it for that time. We've written all the software that communicates with the booking system, and if you don't have a booking, it'll boot you off. It's smart enough to know that your lab mate has booked it and you're in the same group, so that's okay. There's been lots of scope creep and version control, versioning of that. We use just remote desktop to access the VMs. The reason we do that is some of the stuff we tested. So we tried Horizon View. That had a lovely feature of even if I was booked and logged on and someone else accessed it, I got booted off and we couldn't change that. So that was kind of not desirable. We tried VNC. That was kind of horrible because you'd lose the cursor, you'd get keys not mapping properly. And there was a bit of a security issue because someone could VNC over the top on someone else's session that they had authenticated with their university credentials. So we just use plain Windows RDP. Was there a third question?
There are a couple of them. So also, Julia was asking about your choice of static versus dynamic VMs. So why did you choose? What is your reasoning for choosing the static version?
I want to say it's easier with having Windows. The IT teams kind of said don't do Windows dynamic VMs. And that I think answers one of the other questions that we run for our infrastructure Windows because we still have Zen and LASX and the kind of offline analysis tools. That all needs Windows. What was the other part? Sorry.
So this was for the static versus dynamic.
So yeah, the static is just it's easier.
Another question from Patrick is about the licensing. So he's saying if you have only one machine license, you can host it only. You can host the software only on one virtual machine, right? Or how
So
does it work in general? Like if you have these.
So the way we do it, yes, that's how it's done. It took a lot of convincing to a number of vendors because the way it often can be done is that you can have one license to multiple VMs, and that would obviously breach the end user license agreements and things like that. So we have convinced a number of the vendors who don't actively say that they can do remote on their licenses that we have protocols in place. And as I said, we track by the minute which software is running. So if any time they ask, we can show them and have a kind of a proper audit trail from that. That also plays into not needing the dynamic ones because we can monitor the usage patterns and go, no one's using more than a terabyte. So we can kind of shrink that box down and have a smaller one and just, yeah.
There is also a follow-up question on the virtual GPU. So Patrick is asking whether you have instability issues or whether it works okay for you. So for example, that one part of the GPU could jeopardize the other parts of the GPU that are running other jobs. Does it happen to you or?
So that could happen if you oversubscribe the total resource pool. So I do know of one facility that was doing that where, they only had, say, a finite amount of resources and they had double that you could book. And they were just hoping that no one would use everything all the time, sort of like airline ticket sales or ISP internet providers. We didn't like that idea. So everyone gets their own thing. What we have seen is one of our generations of the servers we bought and got greedy and we went for way more cores. So we had, I think it was the Milan AMD where there were more cores, but they were slower. And that saw pretty detrimental effects. So not many softwares make use of multiple cores. So now we will spec it with less cores, but faster.
Okay. So I think we can also move to the other questions, to the other topics. So I think there was a question regarding the image processing portals, whether you can also use it for image analysis development. Like, I guess, for example, if you want to create macros or maybe write scripts or even develop software, but it's just to run actually the analysis?
So the image processing portal was kind of geared to an audience that wasn't developing stuff, because if you're at the level of like writing your own full Python Jupyter notebooks, you can probably use the Open On Demand or your own things and can navigate Slurm on your own. So this was more as like a um, student who's got multiple terabytes spread over thousands of time points of volumes. They just want to MIP it or just do a processing step or deconvolve it and then do scripts on that stuff. It was kind of built with that in mind. Um, so yeah, they're kind of two separate circles I see in our use case. So.
Makes sense. Next question was about the data mining. So Sadib was asking whether it's scalable across different universities and how hard was the deployment to if you have varied IT infrastructure across the different institutes.
Yeah, so we leverage some of the features of PPMS, but it doesn't require it, right? So we can put in like a unique identifier in a booking comment and then from the tracking session, and then we can marry that up. That way we can see information about which group they belong to, and that's stored in PPMS that isn't in Active Directory in Windows and in the university system. So we get some extra stuff. But as far as like date, time, username, whoever's logged into the computer, and then it's just whatever software that's running that you can see as a service or an application in Task Manager, essentially, that's what can be tracked. And then that's just going to a Google Sheet. So it's relatively transferable. And yeah, it's interesting to mine, I'd say. We've got some pretty interesting insights with like we'll run a workshop and we see a change in more users using it or something. So that's been nice.
Okay, so maybe one last question because we are
Sure.
slowly running over time. So Beth is asking about how might one collaborate with you on the metadata ontology, because I guess that's a problem of everybody.
Yeah, reach out. I'm happy to do stuff. As I said, I kind of masquerade as a vibe coder. I'm a cell biologist by training. I spoke with a colleague who recommended they were building something similar in RStudio, and they could link out to the ontologies a bit easier that way. So I'm all ears on how to make it better. This was more just, can we begin the conversation with our users and start to mine stuff where we can, but happy to work with anyone who wants to do stuff with it.
Do you see a lot of people using the Remind tool that you created, or?
I would say our kind of casual users who just come in and want a picture, not so much, but the heavy imaging groups are certainly using it because they can kind of see the benefit because they're on the systems every day and are generating a lot of files and things like that. So having that information has been useful.
Okay. Well, thank you a lot. I think we reached our time limit. So there are still more questions. So if you are fine with that, we can pass them to you after the meeting and then we can answer
I
them.
will get to them over the coming days. Probably not tonight though. Thank you.
No worries. Thank you so much. Thank you everybody for joining
Thank you.
this seminar, and we will see you next time.
Thank you.